Hi, my name is Stephen Chan. I'm a physician scientist and professor of medicine at the University of Pittsburgh School of Medicine. I also direct the Vascular Medicine Institute and the Center for Pulmonary and Vascular Biology and Medicine. In this video, I am excited to tell you about our lab's most recent discovery, examining frataxin deficiency and its role in promoting endothelial senescence and pulmonary hypertension.
Pulmonary hypertension describes a progressive and multifactorial disease of the lung vasculature. Patients are classified into several clinical groups based on presumed etiology and histology. Now, the majority of research and our limited therapeutic options apply to the most severe subtype, Group 1 PAH. However, both Group 2 PH due to left heart disease and Group 3 PH due to hypoxic lung disease have been increasingly prevalent with very little understanding of the molecular drivers of disease. Now, turning to what we do know, pulmonary endothelial dysfunction is a defining feature of vascular remodeling in PH, but less is known about the cell type's dynamic and spatiotemporal phenotypes important to disease. Notably, in the pathology of peripheral vascular beds, DNA damage and cellular senescence are prominently seen. Senescence is a state of stable growth arrest, and while this pathophenotype has been linked to PH due to congenital heart disease or chronic lung disease, its relevance in other PH groups is unclear. It's also known that senescent cells produce an assembly of factors referred to as the senescence-associated secretory phenotype, or SASP factors, that promote tissue inflammation. In cancer and diseases of aging, senescence can be traced back to pathologic DNA breaks that can lead to a DNA damage response. If this damage response is compromised or overwhelmed, senescence can result. While genotoxic stress has been observed in patients with PH, the complex regulatory events that control endothelial senescence and how such events fit into the development of pulmonary vascular inflammation and remodeling are not known. With this in mind, our laboratory has previously demonstrated iron-sulfur cluster deficiency as a key driver of PH. Iron-sulfur clusters are evolutionarily conserved bioinorganic cofactors required for redox reactions across the cell, including mitochondrial respiration and DNA replication and repair. Frataxin, abbreviated here as FXN, is one of over 30 assembly proteins required for appropriate iron-sulfur biogenesis. Genetic frataxin deficiency, driven by trinucleotide repeat mutations, causes a disease called Friedreich's ataxia, a multi-system syndrome in which mortality is largely driven by hypertrophic cardiomyopathy. And in general, hypertrophic cardiomyopathy is accompanied by pulmonary hypertension, which is thought to result from left ventricular stiffening. However, that may not be the entire story. In 1963, a single case report was published describing pulmonary vascular remodeling in a patient with Friedreich's ataxia. Now, little attention has been paid to the study of lungs in Friedreich's ataxia since then, but these data emphasize that PH could constitute an important feature of the syndrome independent of or additive to the left ventricular afterload seen in hypertrophic cardiomyopathy. Therefore, we hypothesize that frataxin deficiency, due to either genetic or acquired triggers, may disrupt iron-sulfur dependent DNA damage pathways, thus resulting in senescence and promoting pulmonary hypertension. Now, to determine whether cellular senescence develops in frataxin-deficient endothelial cells in PH, we quantified vascular expression of frataxin and the senescence marker called CDKN2A or P16 in multiple in vivo models of PH representing diverse clinical subtypes. As an example shown here, immunofluorescent confocal microscopy revealed frataxin, illustrated by gray in the healthy CD31 positive endothelium in the column on the left, was markedly reduced in pulmonary vessels of patients with Group 1 PAH, shown in the middle column, and Group 3 PH on the right column. The same was found across other animal models of Groups 1, 2, and 3 PH, as well as from cultured pulmonary microvascular endothelial cells directly derived from PAH patients. Now, to determine whether senescent cells are distinct from other known endothelial phenotypic cohorts known to drive PH, such as proliferating endothelium, we analyzed single-cell RNA sequencing data from 3 human lungs of pulmonary arterial hypertension patients compared with 4 non-pH controls, matched for age and absence of senescence-altering medications. When comparing endothelial cells specifically and using an expression cutoff that distinguished high versus low levels, mean percentages of senescent P16 positive green cells and proliferating MKI67 positive cells shown in orange were both increased in endothelial cells of PAH lungs shown in the right as compared to no PAH shown on the left. There were only rare double positive cells that were identified. Notably, cells displaying high frataxin levels shown here in orange were also consistently P16 negative, while P16 positive endothelial cells shown here in green rarely displayed such high frataxin levels. Thus, we identified a cohort of P16 positive senescent pulmonary endothelial cells without substantial frataxin expression, distinct from proliferating endothelial cells in the same lung. These data together indicated a potential mechanistic link between frataxin, senescence, and pulmonary hypertension. In data I do not have time to show you but will only summarize, we found in cultured PA endothelial cells that acquired triggers of pH, like hypoxia and inflammation, downregulated frataxin via a HIF-alpha-dependent epigenetic pathway. Downstream of that, given the importance of iron-sulfur clusters in nuclear replication and repair proteins, depicted here on the left, we demonstrated that acquired frataxin deficiency disrupts the iron-sulfur dependent nuclear activity of PA endothelial cells, leading to replication stress, DNA damage, cell cycle arrest, and ultimately senescence. Independent of acquired causes of endothelial frataxin reduction, we sought to establish the relevance of endothelial dysfunction to pulmonary vascular remodeling in Friedreich's ataxia. Via rare lung autopsy samples of 3 Friedreich's ataxia individuals, histology revealed significant pulmonary arteriolar remodeling in the medial and adventitial layers with indications of inflammatory cell infiltration, strengthening the principle that Friedreich's ataxia and pH are linked. In an effort to characterize frataxin-dependent endothelial function in the context of Friedreich's ataxia, fibroblasts were collected from these same patients with known frataxin mutations, a 30-year-old male and a 36-year-old female. These fibroblasts were reprogrammed and differentiated into inducible pluripotent stem cell-derived endothelial cells. And similar to our prior results, these cells exhibited features of DNA damage, growth arrest, and P16 expression, demonstrating that genetic mutation similarly drives senescence. To model this in vivo, we investigated endothelial frataxin knockout mice exposed to chronic hypoxia. This allowed for mice to exhibit increased DNA damage and P16 transcript expression in the pulmonary endothelium. And in data I don't have time to show you, an inflammatory SAPS phenotype was observed that promoted elevated accumulation of CD11b positive interstitial myeloid cells in the pulmonary vasculature and increased pulmonary vascular collagen matrix deposition, both mirroring the adventitial remodeling seen in Friedreich's ataxia lungs. Importantly, this drove pulmonary hypertension at the hemodynamic level with increased right ventricular systolic pressures and increased Fulton index, which is a marker of RV hypertrophy. Importantly, consistent with single-cell RNA sequencing data from human PAH lung, an inverse correlation shown on the left between frataxin expression and P16 was observed. This was accompanied by a positive correlation between P16 and right ventricular systolic pressure. Thus, these results together emphasize the pathogenic role of the frataxin-deficient senescent subpopulation in pulmonary endothelial cells, promoting vascular inflammation, remodeling, and pH. Finally, we sought to explore the contribution of frataxin-dependent endothelial senescence to the development of multiple pH subtypes in vivo. To do so, we wanted to determine if pharmacologic clearance of senescent cells by the BCL2 inhibitor ABT263 could reverse frataxin-dependent disease in a variety of group 1 and group 3 mouse models of PH that we had found were driven by frataxin deficiency. For example, the hypoxic interleukin-6 transgenic mice, which are a model of group 1 PAH, were treated with ABT263. While upstream frataxin remained depressed in this model, we found that senolytic administration reduced immune cells in the vasculature, reduced vascular remodeling, and reduced consequent hemodynamic manifestations of PAH, thus proving the crucial role of senescence in halting the pathogenic cycle connecting frataxin deficiency to this disease. So in summary, via discovery platforms spanning